My name is Melissa Goodman, and my modeling project is on synaptic communications. Synaptic communication is a process of communications between two nerve cells. Here is a neuron. The cell body contains the nucleus that has dendrites. The cell body continues into the axon. The axon has a sheath covering of protection or insulation called myelin. At the top of the axon splits into several axon terminals. These axon terminals are what creates the synapses with other cells. When one neuron is connected or creates a very close space between the second neuron, the passing of the signal from a neuron to a second neuron is what we refer as synaptic communications. An action potential, which is the signal electrical current, starts at the base of the cell body from a resting neuron. The intercellular fluid is high in potassium ions and has a resting potential of -70 millivolts. When a stimulus is introduced, initiates an action potential should start generally depolarizing. This is when the stimulus generates the action potential enough to open the voltage gate channel. The opening of this channel gate is called the threshold, which is around 50 to -60 millivolts. Sodium ions rush from extracellular fluid into the intercellular fluid, which reaches a +30 millivolts, and sodium channels close. The cells repolarize and opens the potassium channels to move the potassium from the inside to the outside of the cell. The action potential travels down the axon due to solitary propagation. Propagation or conduction flows similar charges. Solitary propagation is what the neuron uses. The nodes of the axon are able to connect and pass the action potential down the axon. The nodes connect the action potential and the local current skips into the internodes, jumping from one node to the next, and this is how action potential is traveled. When the action potential finally reaches the axon terminal, this is now considered the presynaptic neuron. This presynaptic neuron takes the action potential or nerve impulse and makes neurotransmitter, passes through the space or synaptic cleft. To move to a receiving neuron or postsynaptic neuron in the dendrite, the pulse opens the voltage gate of the calcium ion channel. The neurons will engulf the neurotransmitter that has made sacs. It will then dump the neurotransmitter into the synaptic cleft via exocytosis. The time it takes for the neurotransmitter to leave the presynaptic neuron to the postsynaptic neuron is called the synaptic delay. Once the neurotransmitter reaches the postsynaptic neuron, the bond to the iotropic receptor, aka the Vanguard gate ion channel, and opens the gate. This means the neurotransmitter is what directly bonds to the receptor, opens the gate by changing the receptor's shape. This will then continue or stop the impulse depending on the neurotransmitter, and the exoterotransmitter will cause depolarization and continue the action potential. An inhibitor neurotransmitter will hyperpolarize and stop the action potential.